From Microbe TV, this is TWiP, This Week in Parasitism, episode 169, recorded on April 9th, 2019. I'm Vincent Racaniello, and joining me today here in New York City, Dickson Despommier.
Hello there, Vincent.
And from a remote location, Daniel Griffin.
Hello, everybody.
Hello, Daniel.
Got cloudy.
Yeah, it did. It's supposed to rain later.
Rain is good.
Rain is always good, unless you live in Nebraska.
I put seed down 2 weeks ago.
Yeah?
And it's just sitting there, and the birds are eating it because there was no rain.
Nice. That's
I
nice.
said to my wife, why isn't it germinating? And she said, it needs water.
She happens to be right.
We actually have a follow-up before we get into our case. From Lucian, Dear TWIPsters, I wanted to point you all to a recent paper identifying succinate as at least one signal sufficient to activate tuft cells and trigger a type 2 immune circuit.
Wow.
In fact, providing succinate in a mouse's drinking water before infecting it with Nipostrongylus brasiliensis will shorten the duration of infection.
That's remarkable.
Full disclosure, I worked in a small way on the paper provided as a rotation student. Cool. It's tantalizing to think that rather than detecting the equivalent of PAMPs, type 2 immunity might rely instead on deviations from metabolic homeostasis to detect pathogens. As always, thank you for a fantastic podcast.
That would be cool.
So this is a paper in immunity. Detection of succinate by intestinal tuft cells triggers a type 2 innate immune circuit. What do you think of that, Daniel?
I think this is fascinating stuff.
I do too.
And I was going to I realized that probably not everybody knows what PAMPs are.
So, just little diapers that you put on little kids, right?
I thought we should mention these are. Actually, I like the term PAMP as opposed to DAMP. So these are pathogen-associated molecular patterns. And, because usually our immune system is using a two-signal system. So you're using one detecting a pathogen-associated molecular pattern, and then you're using a second one to try to determine, is this a pathogenic situation, or as in most of our interaction with potential pathogens turn out, is this just a symbiotic or a commensal or something along those lines? So, and I think this is great because, as we always talk about with our swords into plowshares, as Dickson coughs over there, nothing to cough.
Sorry, I can't help it. You asked me to leave, I'll leave. But
Goodbye.
Just stay with us. Don't
a lot of editing out there, Vincent.
Goodbye.
It's all good. It's all good, Dickson. No, but as Dickson, I think sort of proposes the turning swords into plowshares, we would love to find out different things about the parasite-host interaction and capitalize on them without necessarily filling ourselves with potential pathogens.
Right.
And I like the idea that maybe you can use something fairly innocuous, like the succinate, to sort of rev up our GI taste cells so that they can sample and give us a better sense of what's going on without having to put a full microbe in there.
Yes.
So the idea here is that the succinate is produced by the parasite, and that is detected. And what is succinate a product of? Does anyone remember?
It sounds like it's aerobic metabolism.
Succinate is a dicarboxylic acid.
Yeah.
And what does it do? In respiration.
Yeah.
Here we go. Let's read it. Right?
Yeah.
It's in the TCA cycle. It's a key intermediate in the TCA cycle, a metabolic pathway used to produce chemical energy in the presence of oxygen.
Yes.
It's generated from succinyl-CoA by the enzyme S-CoA synthase in a GTP-ATP producing step. Very cool. So if you remember the circle of the TCA cycle, it's about at 7 p.m. is succinate.
Yeah, it actually down lower left there.
Yeah,
that's right.
right, sort of shooting out into electron transport chain, right below fumarate.
Remind you of time is running out.
Time is running out. Yeah.
Yeah.
Thank you, Lucian. That was beautiful.
Yep.
All right.
We happen to have an expert on the tuft cell here at Columbia.
Who's that?
Michael Gershon.
Oh, and wouldn't it be cool if his name was, or her name was Michael or Michaela Tuft?
No, it would have been cooler if he had gone to Tufts. But we might want to invite him on the show sometime to give us more insight about the Tuft cell structure and function.
Sure, that's a great idea.
Yeah, he was one of the authors on the last paper we reviewed.
Yes, I remember that. All right, Daniel, let's get back into clinical cases. What do we got here?
Yes, for everybody tuning back in and those tuning in for the first time, let me remind and acquaint everybody with the case from TWiP 168. We discussed an 18-month-old child, and this 18-month-old child had been on a medical mission with his parents and an older sibling back in the U.S. for, we said, somewhere in the 2 to 3 month range. And just as they were leaving this remote location where they had been on this medical mission, they had taken a dose of albendazole. The child was at daycare, and the call comes to the mom that the child had passed something. Actually, it was during I add a little bit. It was during the diaper change. They opened up the diaper, and there was this 5 to 6 inch long, round, serpiginous thing that was actually moving a little bit. I got to see the video, which I thought was great. The child was otherwise fine. And I didn't say this at the end, but Dickson was fishing a little. He was like, was there something that happened right before this? and so I'll just give this to people that maybe this will be the people tuning in for the first time. We'll pause it now. They had given the child a second dose of albendazole a day or two before this had happened. Mm-hmm. So, all right, I will give everybody that. Dickson also wanted to know color, things like that.
Picky.
I wouldn't tell.
It's all right. We're armed and ready.
All right, the first guess is from Sarah, who writes, "Hi all, I sadly have zero time for a lengthy analysis of this week's case, so this will be short and sweet. I'll roll the dice on Toxocaracanus or Ascaris lumbricoides. Fingers crossed, looking forward to the next episode as always. Sarah, currently in Sydney."
Dickson, you were shaking your head. Why'd you shake your head? Well, not
for the first one. For the second one.
I just think Sarah should have taken an hour of sleep away and done that. Don't you think?
Well, first of all, the adult of Toxocaracanus is not as long as Daniel described. And second of all, people don't get it. The adult stage of Toxocara, they get the larval stage.
Boy, you're amazing, Dickson.
Occasionally.
You should be a hero.
No, stop it, Vincent. You know the life cycles of the viruses, and we know the life cycles of the eukaryotes.
You know what we should do? We should do a podcast, and we could get all this knowledge out there.
That could work. That's a hell of an idea.
And Dickson, you could even throw in some entertaining stories here. And think? Vincent, you can keep us in line.
That sounds like a plan.
Dan, Dickson, can you take the next one, Dickson?
I will do my best. Ben writes, "Dear TriTWiPomonads."
No. TriTWiPomonads. 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 Firstly, I need to clearly err in regards to South Australia geography and the pronunciation of Adelaide. Dickson had it right when he said he pronounced it Adelaide. But said that us Adelaideans would pronounce it Adelaide.
Yes.
We do have a somewhat strange accent here, though. I usually get asked if I'm English before Australian when I travel. Adelaide is about 8 hours along the coast from Melbourne, and it is not in the Grampians, but you do have to go past the Grampians to get here. The city of Adelaide is flanked by the Adelaide Hills, Barossa Valley. They grow a lot of great wine grapes there. Clare and Eden Valleys, which makes it perfect location for wine lovers. See? On top of this, we are right on the beach. Where I live now is 15 miles away from the beach, wine country, and my university. So I'm definitely going to be sad to see this place go when I hopefully move for a postdoc in the future. I tried to do my research in this case, but I was really struggling. It was impossible to hear a child had passed a worm of that length and not start thinking of Ascaris straight away. But then the child had taken albendazole, and it was a few months later, and it didn't all sound right. So here are my options. Number one, the serpiginous object objects are Ascaris lumbricoides. This seems a little on the small side for what I've read, but perhaps the worms only get to a smaller size in a child. The possibilities I see for how the child could be infected with Ascaris include albendazole resistance. The child was infected with Ascaris before coming back to the U.S., but the worms were resistant to the drug. If not sure if this is often observed, albendazole may not be effective against Ascaris in some of the earliest stages of infection. I couldn't find anything supporting or refuting this. The albendazole might have been taken sufficiently long enough before coming back to the U.S. that the child contracted Ascaris between albendazole treatment and leaving. Apparently, Ascaris worms can take longer than 3 months to be passed in rare cases, so that it could explain any of these scenarios above. I think what he meant was at 3 months to mature. And number 2, the child had ingested something that is not normally considered a parasite, but has managed to survive through the GI tract and grow into a worm. I think I remember a case a while ago where someone passing worms after eating mangoes. 3, the child has eaten one of the items on the extensive list of things that kids have eaten, and the parasites only find the evidence in their nappies. A little side note, I think you would like the cover of the latest edition of the International Journal of Parasitology. Only full-size image I could find in their Facebook page, and he lists that, is a parody of the Beatles album Let It Be that they've dubbed Let It Babesia. Also a suggestion for a paper in Nature, although sadly behind a paywall, parasitic apicomplexan that commonly infects coral, yet retains chlorophyll biosynthesis genes. Fascinating stuff. Thanks again for podcasting a parasitic spell over me. Regards, Ben.
Would you be interested in doing that paper, Dickson?
Absolutely.
A widespread coral infecting apicomplexan with chlorophyll biosynthesis genes.
I definitely would be.
Let's do that. This Facebook page with the picture, he says, what is it? Let it babesia.
Yes.
So the picture, the journal cover. So do you remember the Beatles' Let It Be album had 4 panels with each of the 4 Beatles pictures? So here they have that, except the first panel is John Lennon. The second panel is a tick.
Right.
The third panel is a face that looks like an alien. And the fourth panel is a cow.
Right.
Can you explain that?
I'd rather not.
Rather not.
Let me open this so I can see what we're doing here.
I should follow this. I'm going to follow them. You can control who can see the people, blah, blah, blah. I'm following.
Oh, actually, I like this, actually. So the tick, right? And we've talked a little bit about this.
Babesia is transmitted.
Not only is Babesia tick transmitted, but the first description of a arthropod vector-borne illness was a Babesia that infects cattle, Texas.
That's right, Lone Star fever.
And the interesting thing, it looks like an alien, but I think this is part of a partial Maltese cross type thing. So when they replicate, they replicate into 4, and they actually, if you were to have 4 of these, but usually you don't see 4. Actually, people say, "Oh, I didn't see the Maltese cross, must not be," but sometimes you might just get a view where you see 2 of them, and it does. It looks like these alien eyes. So this is
I see.
Yeah, this is cool.
What's the cow in there for?
No, the cow was the first
cattle.
The first
shipping fever.
Yeah.
No. It's called Lone Star.
Lone Star. Shipping fever was something else.
Yeah.
What's that?
That's Borrelia recurrentis.
Borrelia.
Yeah.
Bergdorferi.
Yeah, I think so. No, not Bergdorferi. Recurrentis. I think recurrent fever, relapsing fever.
Ben is in Adelaide or Adelaide,
depending on who you talk with.
But what is John Lennon doing there? What's he?
Well, he was a reference to the cover. If they had a 4th image about Babesia, I guess they would have put it there.
Maybe he had Babesia.
Yeah. I presume that if you're producing honey on a common basis and you have another magazine, you could just call it Let It Be, right?
Dickson. Daniel, can you take the next one, please?
This is Nathan. Hello, TWiP experts. This answers to this week's case should be obvious, but there's one piece that makes me question my thinking. I'll start by listing the important details that Dr. Griffin mentioned. 1, 18-month-old child traveling abroad with family. 2, has been back in the U.S. for 2 to 3 months. 3, took a single dose of albendazole before leaving. 4, has been in frequent contact with soil, 5, and the sort of 18-month-old past a 5 to 6 inch roundworm at daycare. Now, based upon the size and shape of the worm, it sounds like the child was infected with the giant intestinal roundworm Ascaris lumbricoides. Dr. Despommier describes this organism as being the size of a pencil, 7.5 inches. This case is curious because a single dose of albendazole should kill adult worms and prevent eggs from hatching. Albendazole disrupts glucose uptake and depletes glycogen stores in the worm. There are a few things that I think could possibly be going on. 1, the child did not take the dose of albendazole, so the worms were not eliminated. 2, the child became infected after taking the medication after returning to the United States. 3, it is possible that the organism is resistant to albendazole and the treatment did not work. There have been some recent studies that have questioned the efficacy of albendazole against Ascaris lumbricoides. For example, reduced efficacy of albendazole against Ascaris lumbricoides in Rwandan school children was published in the International Journal for Parasitology, Drugs and Drug Resistance, 2017. 4, the worm isn't a parasitic worm, maybe a common earthworm, found its way into the soil. Can't believe that I'm even bringing this up, but it was seen in one of the labs where I used to work. It sounds like the family may have to find a new daycare to attend. P.S. I have included an image of our preserved worms that we show our students. I have named them collectively Eleanor. Hey, I also have a question for Dr. Despommier. Yes. When I took a parasitology course almost 10 years ago, the instructor mentioned that he traveled to Portugal with his family when he was young. He said that one of his extended family members was infected with Ascaris lumbricoides and that his family put a bowl of warm water under his chin.
Milk. Warm milk
under his chin.
That's
and right. The worms migrated out of his mouth. I found that the imagery was distracting. I recently looked up using a bowl of milk as a treatment for Ascaris lumbricoides online, and as I expected, there isn't any information available. That's my instructor telling a tall tale. Have you ever heard a similar story, Nathan?
I could go on for hours about this one, but I will just say that your instructor was pulling your leg, and they used to say this story for tapeworms, and they used to say things like, teasing it out by holding a cookie near the anus of the patient so the parasite could follow the scent of the food. The parasite would then come out. This is a similar story, and they're absolute.
You could talk for hours about this, Dick?
Farces, because every worm has its own little nuance about what you should do in order to get rid of it. This is not going to work.
No, it is funny because I remember being taught these things by my elders that with the parasitic worm was so they would starve you, like you didn't get to eat for a few days, and then they would hold food in front of your mouth, and the tapeworm would come up, and then it would come out of the mouth to grab the food, and then you'd grab it. I mean,
pretty disgusting.
These are the horrific stories that if I didn't behave well, I would get a tapeworm, and that was how I would have to undergo my cure.
Right. So thanking all of those people for putting us in a psychotic episode at a young age.
Yes, I will behave, I promise.
Exactly right. I'll do anything. I'll eat my spinach.
All right, Lucian writes, Dear TWiPsters, the only parasite that I can think of would be 5 inches in length and round is Ascaris. As to why the child was infected despite treatment with albendazole, there are other periods in the parasite's life cycle where albendazole is less effective. I could find no mention of this, but it strikes me that perhaps while the parasite is in the lungs, or perhaps while it is in the liver, concentrations of albendazole may not be sufficient to kill the parasite. In fact, I'm not sure you'd want the worm to die in your lungs. Since maturation can take months, this could explain the delayed discovery of infection after treatment. Many thanks for all you do. All right, we have one more, Dickson.
Yes, sir. Dear...
No.
What do you mean no?
Right, who wrote it? Read who wrote it.
Oh, Dr. Anderson's parasitology class of North Greenville University writes.
Very good.
Dear TWiP... Is that an I or an L? It's an I.
It's got to be an I, Dickson.
Dear TWiParasites. It's a stormy day in Tigerville, South Carolina. Our parasitology professor is a big fan of your podcast. Well, I hope you are too. Below, we have compiled a class response to the case study presented in the last episode. We first considered identifying the parasite based on the exposure the patient had had in remote location outside of the U.S. Using the information provided by Dr. Griffin related to the size of the parasite, we narrowed it down to Ascaris lumbricoides and Wuchereria bancrofti. Considering Ascaris. A bullet point. We know that this helminth is endemic in poverty-stricken areas, such as the one described in the case. Another bullet point. Further, considering the day-to-day activities of the child, the embryonated egg would most likely be ingested when the child was playing in the dirt. Third bullet point. The size of the organism the daycare worker encountered aligns with the average size of an adult male Ascaris worm, being 5.9 to 11.8 inches. And the final bullet point, the description of a roundworm rather than a flatworm also led us to this conclusion. Okay, now, considering Wuchereria, which is a more difficult way of putting this worm in context. First bullet point, this worm is also a roundworm and can grow up to 5 to 6 inches and take 3 to 6 months to develop into a mature stage. However, since this infection typically involves the lymphatic system, we were able to eliminate it as a potential diagnosis. Considering the unique presentation of the case with the delayed appearance of the worm, we have researched a few options to try to explain this outcome. First, bullet point. First, low parasitic loads are usually asymptomatic as seen in the patient, so the medication could have been partially effective. Second bullet point. We also are unsure if the dose provided to the child under 2 years of age would be effective to eliminate the infection. Fourth bullet point. Typically, as noted in the study by Antigens et al. In 2014, one dose of albendazole is effective at clearing 85% of adult worms. However, we are curious if the treatment is not effective at killing the migratory larval stages of the parasite. Final bullet point, this would lead to an adult emerging 2 to 3 months later, post-treatment, which may explain the organism encountered by the daycare worker. With the information provided in the case study, we don't have a clear explanation and are still thinking about what would have caused the immediate evacuation of the worm. We considered that perhaps the child had developed a fever or some other illness that is causing the parasite to evacuate the premises. We hope to win the book. It would be an excellent resource for our school. Stay parasite-free, Dr. Anderson's parasitology class of North Greenville University, Tigerville, South Carolina.
Wow.
Well, we're going to send you a book anyway. That's what I'm going to say. Right now, I can unequivocally say that regardless of whether you win or lose, you will get a book from us, and we will sign it.
Why is that, Dickson?
Because it's a school, and they deserve this book, because then many, many students can take advantage of this gift. And we give our book to schools anyway, so we should give a book to the school. That's my opinion.
So that's our list for this time.
It is.
Guesses, yes, because it was a short turnaround here on TWiP.
Exactly.
So, Daniel, what do you think?
Now we get to hear what you two might think and other questions and thoughts. So let's start with you, Vincent.
So I remember we do a case where it was an earthworm?
We might.
I think we talked about it. I don't know if we actually did it as a case.
No.
Wasn't there a story where there was someone, it was like a construction worker or something? But I don't know if we actually did it did we do it as a case? I hope not.
I don't remember. Well, there's one way to find out. We can search for TWiP and earthworm because it could be in the keywords.
Could be.
Yeah. I actually keep an Excel spreadsheet of all the cases that we have done.
Good, man.
If you can imagine that.
Incredibly organized.
No. We didn't do but there is one letter which talked about visceral larva migrans associated with earthworm ingestion. You shouldn't eat earthworms because you could get larva migrans from them.
Yeah, actually, there was an episode on Long Island, I remember, not so many years back. It was one of these dare things with a few children actually daring one another. I don't know if you remember this story, but there was a girl and 2 boys. I remember. I think I remember that because I could see boys eating worms. I usually think of my daughters as being smarter than that. But is it sexist? I'm not sure, but I don't think they would eat an earthworm. But my son, on the other hand, yeah, he might.
I see.
So
it's like
yes, please don't do that.
Well, I thought because of the size that it would be an Ascaris, but I don't understand why it's coming out 2 to 3 months after the last dose of albendazole. Of course, it could be that there were other doses in the interregnum, as we say. You know what the interregnum is?
I couldn't possibly guess.
So my guess would be Ascaris only because of the size.
Mm-hmm. Okay.
But that's a puzzle why it took 2 months to come out.
What are your thoughts there, Dickson? Do you want
I brought my answer with me, and I'm going to just show it up to the camera.
Yeah, we got it.
Dickson, you going to take your picture?
We have a bottle of Ascaris worms, and those were yours, right? Those came out after your trip.
They didn't come out after me. No, they were a gift.
Very impressive, Dickson.
Gift.
Well, I will say that was my diagnosis as well in this case, and I actually got to see the video, so I got to see the worm, and it was very recognizable as an adult Ascaris. But part of why I wanted to present was this interesting question of how does this happen? As we saw from the case, this young individual took this albendazole before they left their remote location, and the thought was, isn't that enough? Shouldn't that sterilize them? How can they end up with an Ascaris coming out into the nappy, 3 months afterwards? And should I comment? Do you have comments, Dickson? Do you have thoughts?
I have lots of thoughts, but I don't want to step on your thoughts because you're the doctor on call. So you can give a longer explanation as to why.
I'll go first, and then you can jump in, and I'll bring up some of the things that our emailers wrote it, which I liked, and I actually think it's great when it's sort of the emailers are jumping in with here the couple of things. So the first thing I like to point out is the data that was presented when they discussed the Rwandan school children, is that when you take a dose of albendazole, it's not sterilizing. You will not kill 100% of the Ascaris all the time.
Exactly.
So that's enough. That could be the explanation.
Right.
And the reason you're doing these deworming campaigns is, as we talk about with most of the soil-transmitted helminths, is it's a parasite load that creates a lot of the morbidity and problems. Now, there can be a few exceptions. We've talked about how Ascaris can have aberrant migrations. An Ascaris worm might migrate up into the biliary duct. And there are thousands. I think the estimate is 10 to 30,000 cases of that a year, what actually causes obstruction and infection in the biliary system. It also can migrate into the appendix, which can actually cause a blockage infection there. So aberrant migration is an instance where a single Ascaris could cause problems, but usually it's a load with most of the soil-transmitted helminths. So when you do your mass treatments, et cetera, you're getting the load down, but you're not necessarily getting 100% sterilization. The next thing people brought up is, does it treat every single stage? Like, what's this story of a second treatment?
That's important.
And there is a little bit. How do we think albendazole works? And we think it binds to tubulin. So we think it actually affects the cytoskeleton, and that can affect several things. And it was interesting. I mean, I maybe I have an empathy towards parasites, but you could see this worm was not moving quite right. It was moving, but it didn't have the nice movement that I would hope a healthy Ascaris was crippled.
That's right.
So this poor worm was unable to basically swim against the fecal flow and came out. Now there is an efficacy of albendazole against adults as well as some of the earlier stages, but I'm not convinced that it can really be as effective against the eggs. So I think that, if you took this, right as you got on the plane, okay, maybe, but there's always the chance that there's a stage early on, the egg early in that you don't have. So I think someone brought up the two is how effective is albendazole about every single step. Or
the migrating larva.
Yeah, or the migrating larva. I think it has an impact on the migrating larva, but not going to have the same efficacy as an adult, which is basically going to be unable to survive and get passed out. So that's actually why I think this person was doing the protocol of the one dose before you leave and then undose again the 3 months later to get even a further reduction.
So let's take this one step further. And then obviously you're the physician and you see this worm and it's a small child and they've received a dose of albendazole and it's 85%. Let's assume it's 85% effective. You're going to still think that there might be some more worms there. So you're going to have to do a stool examination, right? To follow up to make sure that there aren't worms there. Or if there is, it's a male and you're going to be out of luck because that's the last worm that didn't get treated, and unfortunately, this person then developed a migratory phase of the infection, and that's been recorded, by the way. So, all these possibilities are real, but I think I totally agree with what Daniel was saying, that this is a clear-cut case of 85% were treated, and by the way, this was the worm that was found. But if there were 20 worms and 19 of them passed in a way that did not allow them to be observed, like if this kid was potty trained, for instance, or toilet trained, you could have missed that event and still had one more worm to go, and then out it came into the diaper, right? And so there you go, because when you say 85%, you can't be talking about 2 worms or 4 worms. You have to be talking about many more than that. So I think, everything falls into line with what we already know about Ascaris infection, both in young children and in adults as well. So I'm very comfortable with my diagnosis.
Yeah, it's an interesting issue, right? Because someone brought, maybe they got infected after they left, and it's so as we've talked about before, I'm going to go into why I don't think that's the case. Is well, what about the daycare workers? Oh my gosh, could they have got infected with Ascaris? And there's a really interesting issue that I would say a lot of our listeners are probably getting acquainted with, but the concept of that it takes a period of time for the eggs to become infective or to embryonate,
and they have to embryonate outside the host.
Yeah, so they have to get out. They have to get into an appropriate environment, and then it takes several days. So it's just a question of hours, like pinworm, in the soil, and then they have to get into a new host. The person changing the diapers is really not at risk, it's not common that we see this in in places that actually have sanitation systems.
Sure, but they could have been infected as they left.
That is completely true. I mean, they were still in the environment, and as I mentioned, they didn't necessarily take it on the plane.
Right.
Like, all right, let's wash everything.
Right.
Jump on the plane.
Exactly right. It's a very common infection among Peace Corps workers and people living close to, like anthropologists and that sort of group of people. They tend to pick these things up regularly. And it's a good drug. I mean, the drug works very, very well against the adult stage, but you got to take it twice or maybe even 3 times with stool exams in between to make sure that there are no more worms than there are passing eggs.
Yeah, if you're trying to get to that level. And the interesting thing about Ascaris is probably one of the most sensitive, where they say with the other soil-transmitted helminths, particularly whipworm, you probably want to do 3 doses to get to a higher level.
That's right.
So that's – and actually sometimes when we'll see a child who has what we think is morbidity from such a heavy helminth infection, we won't just do a single dose.
Right.
We'll do 1 dose, we'll repeat the dose, and what you're really trying to do is 85% and then 85% again, really trying to get that burden low.
Exactly. The thing to say about Trichuris is that it's got a tissue phase as an adult, and it's probably very hard to achieve a therapeutic level of drug by just a single dose. It has to accumulate in the tissues in order for it to be effective.
Yeah.
Daniel, remind us, how did you get to this case?
Oh, so this actually was the child of a colleague that I had worked with, so that was why I got to see the video.
Oh,
right.
But did you participate in the treatment of this patient?
So there wasn't actually treatment beyond this. Actually, they had taken the first dose, and then they took the second dose just a day or so before this happened. So it was more of the curiosity. They were, the people at the daycare place were not. I don't think curiosity was their response, but that was a response by the rest of us to say, "Oh, hey, we know what that is."
So you assume that the second dose was the one that caused the worm to be evacuated.
I think so, because it looked a little bit, that was probably the trigger. But no, I think someone else brought up the others. Sometimes we'll see someone gets a fever, they don't feel well. I actually had a friend that I had traveled with, and they came down with something, and that was actually what triggered the Ascaris to evacuate. So
got it. They're lucky that it came out that way rather than the other way, aren't they?
Yeah, because they will. The Ascaris will try to flee, and we've seen them come out people's noses, etc.
That's right.
Not good.
Yep.
All right, shall we give away a book?
You bet.
We have 5 guesses, number between 1 and 5. Are you ready, Dickson?
I am.
Number 4. I don't know if Lucian has won before, but it's Lucian. Winner.
It is Lucian.
And Lucian, please send me your address at TWiP@microbe.tv. And if you are overseas, that is outside of the continental U.S., please include a phone number.
Right.
Because I need that for the package.
Right. And we are sending one, I think, gratis of Parasites Without Borders to the...
that can be a plain one, doesn't have to be.
Yeah, that's right. That's right.
So to the class, to Dr. Anderson's parasitology class, Dickson, Daniel and Dickson would like to send you a book.
We would.
So please send an address, TWiP@microbe.tv.
Exactly.
Now, did everyone get the right?
Well, everybody gets it.
On.
I mean, they mentioned some other parasites that weren't quite possible, like Toxicaricanus and Wuchereria bancrofti, but they all stuck to their guns and said it's likely to be Ascaris.
Dickson, did anyone ever give you a sample and it had an earthworm in it because they were joking around?
No, but in the laboratory, while I was a technician, we do have something called mucus casts.
Yeah.
And mucus casts can often look like worms.
What is a mucus cast?
That's exactly right. Daniel, would you like to explain this?
I'll explain it, but at some point I'm going to present a case of mucus cast. Just warning people, we're gonna wait a few months, but it's interesting. They'll bring in stuff and they'll look at this, and let's say they're about the length of Ascaris, right? I've seen these where they're 6-7 inches long.
That's right.
They tend to be pale.
Yeah.
When you mess with them at all, they just sort of fall apart because what they are is mucus that is basically from the inside of the colon, but they're long, they're a little serpiginous, usually not as serpiginous. They're somewhat flat, so they're not round, and they fall apart. There's no structure to them.
That's right.
But people bring them in not because they're being, troublesome or anything, but they're actually worried. They look at it, they say, "Oh my gosh, I've got these in my stool," and they look to them and you look closely and you realize, oh, they're just mucus casts.
That's right.
So how do we know that they're mucus casts? Well, you look at them, they have no structure to them. They're obviously not. But they're just mucus.
To an uninitiated person, they certainly ruled that in their mind's eye, at least, that they could have a worm. So we're happy to tell them they don't.
Let's move on to a paper.
Indeed.
This paper is from PLOS Pathogens.
Yes.
Whole genome sequences of the bovine blood fluke Schistosoma bovis supports interspecific hybridization with S. hematobium. And this comes from a large group out of many places. First author is Harold Oey. OEY. Interesting. Last co-authors are McManus and Krause, and comes from Queensland University, Australia, Brisbane.
That's right.
It's also Australia, Dickson, the University, Bielefeld University in Germany, University of Melbourne, an institute in Brisbane, Queen's University Belfast, which is in Northern Ireland, the University of Queensland again, another Queensland. Now here we have Salamanca, the Institute of Natural Resources and Agrobiology, and finally the Natural History Museum in London.
Nice.
Now, Dickson and Daniel, this is about Schistosoma bovis, which they say is a common veterinary problem in Africa, Middle East, occasionally in the Mediterranean region. Okay?
Indeed.
And bovis, I assume the problem is in cows, right?
Got it right.
That's a good one.
That was a hell of a guess.
So let's, before we get into the paper, hey, just because it's S. bovis doesn't mean it just infects cows, right?
This is true.
Yeah, that's true. Like strep bovis, that would infect human beings, right? That's a classic. We see a bacteremia. They've renamed it, of course, but it is often associated with colon cancers, and that's infection in humans. But yeah.
Dickson, they say this is a real problem. Why is it a problem?
It's a veterinary problem because of the same reason that it's a problem in people as well. It causes blockage of pre-sinusoidal capillaries in the liver, and it can cause disease if enough worms accumulate in a particular animal. They can get GI bleeding. There's a whole bunch of horrible things that occur as a result of schistosomiasis.
Actually, they say cattle, sheep, and goats. There you go.
That's right.
You were laughing at me for guessing cows, but in fact...
I didn't laugh at you.
It's okay.
I laughed. That was me.
So they say it's transmitted to these ruminants from freshwater snails. How does that? Do they eat them?
No. The freshwater snails actually shed a stage called the Cercaria.
I see. In the water.
Cercaria actually seeks out the animal.
What are these animals doing in water?
This might be a water buffalo.
No, it's cows, sheep, and goats. Are they drinking the water and the Cercaria gets on their tongue?
They can drink the water. No.
Because they talk about penetrating the skin.
I know. So cattle have to drink water every single day. They're not like the East African game animals that don't have to do that for periods of 3 or 4 days, perhaps even longer than that. So cows have to go down to the water.
Do you think they stand in it?
They do. I know they do.
You know? You've asked them?
No, I have lots of photographs of them standing in water.
All right. So the sheep and the goats also stand in the water.
When they get the chance, because it's hot. These are tropical regions of the
Animals
world.
Feel heat?
Absolutely.
How do you know? Because they stand in water. This is a circular argument, Dickson.
Now you're making fun of me.
No, I'm not. I want to know, how do you know if animals feel hot? I know dogs pant.
Well,
right?
I'm not actually saying that they go in the water because they feel hot, but they like to go into the water. They do.
How do you know they like to?
Because they talk to each other and they say, "Hey, come on, let's go for a
Okay.
dip." So they do. They get in the water, and when that happens, these swimming stages from the life cycle live at the surface of the water to begin with, and each animal gives off a chemical signature of their presence. It's mostly fatty acids and linoleic acid and lanolin. Those sorts of things. Lanolin, that's sheep's favorite oil, basically. And it creates a gradient. And these Cercaria actually follow the gradient to its source. And that's how they find their host. That includes us too, by the way. Mm-hmm. So the stage that infects then attaches to the surface of the skin by 2 little sucker discs and crawls until it finds a hair follicle.
Right.
Then it migrates down the hair follicle, and in doing so, it drops off its tail because it doesn't need that anymore. And at the base, I know it's like you wrote this as a science fiction novel. And at the base of it, there is a capillary that actually supplies nutrients
to the hair. Yeah.
To the hair. And the worm then penetrates through the skin into the capillary and gains entrance into the host.
And then it goes through the circulatory system.
No, it sits in the skin. It sits in the skin
skin?
And it transforms a little bit into something called the schistosomula. And the schistosomula then penetrates back into the capillaries and ends up in the lungs. Mm-hmm. And once it gets into the lungs, it undergoes a remarkable transformation. That's when it begins to cloak itself against the immune system. We don't know the exact mechanism that it employs, but we think it employs lots of different technologies, mainly accumulating host proteins on its surface, rapidly shedding its own proteins from its surface so that it's not susceptible to immune attack. And as a result, this parasite then migrates from the lungs via the circulation into the liver. Mm-hmm. And in the liver, it then seeks out a mate. And this is where the real interesting part of this paper comes in. How could a parasite that infects cows and a parasite that infects humans, how could they possibly come together?
Wait, this mate had also come in by the same route.
It has.
So you have to be at least doubly infected.
Correct.
If you just get one Cercaria, that's it.
This is exactly right. The interesting part, though, is that the same snail host is the host for both the bovis and for the human.
Really?
Yes. So that's unfortunate.
Can you find them in the same snail?
That's a great question. And I don't know, but I wish I could ask the authors of this paper that same question, because what goes on in the snail is real interesting.
What goes on in the snail stays in the snail.
No, that's the point. It doesn't stay in the snail. It actually leaves the snail. But there are stages that are poorly understood called the sporozoite stage, and then the sporocyst stage, and then the daughter sporocysts. And then they give rise to the Cercaria. And all the while, you've got a lot of genetic multiplications and sharing of genomes and all kinds of things that go on here. And it's, like I said, it's very poorly understood. So when the snail gets infected, when the egg hatches and the miracidium, again, seeks out its host, in this case, the snail, it's either a male or a female organism. Even at that point, it's both either male or female. It can't be both.
Okay.
So you need a male from one and a female from another in order for this life cycle. But here's the real mystery. Now the parasite's in a gigantic animal like a cow, right? The liver of that cow is enormous.
Mm-hmm.
How do these tiny little organisms find each other?
They find each other delightful.
They do. That's the joke. But I used to say that all the time. But the point is that each one gives off a pheromone that attracts the other sex to it.
It's pretty dark in there. You need something.
It's very dark in there. But meanwhile, they're chomping down on liver.
Actually, that's a good question. Daniel.
Yes.
If you're in a liver, can you see some light shining in through the skin?
Pretty much no.
No?
And I'm not sure what eyes you would be using if you were a schistosome.
No, but if you were an eyed thing.
If you were an eyed creature.
You don't see any no glow, because sometimes you could shine a flashlight on your skin and you could.
That's true, translucent,
but you don't see it in the liver. Huh?
I guess that's interesting, right? If you're someone's, let's say someone's sunbathing and you're near the surface, is there?
That's right.
It's always degrees of darkness, right? It's almost like
what I'm trying to say, though, with regards to the pheromonal attraction, is those are usually species specific.
So
that's what separates them. But in this case, we've got pheromones that mutually attract different species of the same
apparently,
genera.
right? Because this paper is about recombinance between the two, swapping of genetic information. They call them hybrids.
That happens when the male and the female worm get together, and then you start making
a male
eggs.
of Bovis and a female of
right. That's right.
Hematobium have to get together.
They do.
So that can happen. Maybe it's rare, right?
Well, it's not rare enough to keep this out of the human population, and that's why I picked it. I thought it was.
So if a human got infected with a male and a female Bovis only, would that go anywhere?
Or you can't do that experiment.
Well, you can't do it, but does it happen?
We don't know the answer. Humans, for that reason, we don't know.
Daniel, do humans ever get infected with Bovis?
Yes, rare.
Pure Bovis.
It's rare.
Well, I guess this is what's sort of the issue that we're finding out now is that it used to be that we thought like, oh, you just had one, but then they start having these reports in Senegal. In northern Senegal, you had these children, and they're saying they're hybrids, but it raises, I think, as we're sort of going with this, how do these hybrids form? Are the hybrids forming in another animal, and then only the hybrids are getting into the people, or the people at some point get infected?
Yeah,
and the interesting thing we see from this paper is that the S. bovis has the same adherent proteins, so it has the ability to bind. Like, it's not discriminatory at that phase. It's actually they're retaining the proteins that allow both different schistosomes to get into hosts that are not necessarily their primary host.
But it's extremely rare to find someone infected with pure Schistosoma bovis.
Mm-hmm. Okay.
So that means that there are some physiological, perhaps, barriers for this parasite to carry out its life cycle. But when it hybridizes with Schistosoma hematobium, those barriers are removed and they can carry out their lives.
Well,
and I think I should clarify, because that's the hybrids have been described, but I don't know if there's been a case of a Schistosoma bovis mono infection.
I looked that up, actually. I tried to find some information on that before the show, and I could not find any data on that said that it's common for Schistosoma bovis to infect people. It's not.
Yeah, I don't know if it's been described, actually, is my sort of thought, but, we'll...
Intercalatum, which is another bovine species that sometimes infects people, that wasn't even mentioned in this because it's not like the Schistosoma hematobium genome, and therefore they didn't think it was likely that it would hybridize.
Basically, this paper is about sequencing the genome of S. bovis.
That's right.
Comparing it. And
exactly.
I want to tell you how they do this. It's very interesting. So they get S. bovis eggs from the liver of a cow in 1997 in Tanzania.
Right.
They put these eggs in fresh water. They hatch into Miracidia, right?
Good.
Then they use those to infect snails in the lab.
Right.
The Cercaria then were used to establish these
at
the National History Museum in London using mice.
That's right.
Adult worm pairs were perfused from infected mice and frozen. A mated worm pair from the 5th laboratory passage was used for genome sequencing.
That's amazing, isn't it?
Yielding 1.9 micrograms of genomic DNA. And then they did deep sequencing of this pair. 374 million base pairs of DNA.
11,000
How does that compare,
proteins.
Dickson, to the human genome?
That's not a fair question, Vincent.
How big is the human genome, Dickson, you know?
It's 28,000.
How many base pairs of DNA?
23,000?
3 billion.
No, I was thinking about proteins.
No, DNA, 3 billion. So this is much smaller than human. And then they compare them.
Yes.
And, you know, a lot of the proteins are quite similar.
How about identical?
97% sequence similarity between Bovis and Hematobium.
That's amazing.
It's huge.
It isn't. It's almost
98%
unbelievable.
at the protein level of the predicted proteins.
There was a group of them that they hadn't done the analysis.
And now, the cool part is there are distinct regions with over 99% identity over several hundred kilobases between Hematobium and Bovis. And when they compare Mansoni and Japonicum, they don't find that.
No, they don't.
So they think the most likely explanation is interspecific genetic exchange.
Bingo.
The timing, they said, is not clear, right?
Well, they meant that by geologic timing.
Well, they think it happened several generations ago, so that's not very long ago. So probably in their, it must have been before they got the thing from the cow, because they just got Bovis out, right?
Right.
So somewhere in the cows it happened, but how long ago, we don't know.
Well, they estimated 1 to 2 million years or 1.9 million years or something like this, way before there were humans. There might have been hominids that could have done this.
So this is a good point. Schistosomes originated 15 to 20 million years ago.
Right.
And trematodes 400 million years ago.
Exactly.
And the trematode split was 56 million years ago.
That's amazing.
The divergence between Bovis and Hematobium 1.85 million years ago.
Right.
But we don't know when this hybridization occurred, though. It could have been last year. Could have been 5. No, actually, so the sample was gotten 5 years ago in Tanzania, so it had to be before that. Right?
And then they also had some ecological explanations too, with the advent of agriculture and encroachment, and the increase of humans living near their farms and having access to the same ecological zone of transmission would raise the possibility of a co-infection with both of these.
So this could have happened. So when we first started growing cows,
it
was 13,000 years ago, roughly.
Earlier than that, probably
15,000.
Yeah, maybe.
And so it could have happened in the meantime.
Almost all domestic animals were in Africa. Almost all of them came from Africa.
Right, because they were originally wild animals, right?
That's right.
And I presume they were bred to some extent.
Yeah.
And then we started collecting them in large numbers, and we were husbanding them.
We were.
Doing husbandry, and so then we got their parasites, right?
That's right.
Although in this case, I don't know how the co-infection would happen.
Well, the other thing that's interesting to think about is that, remember I said that the Cercaria seeks out its host. Human beings and cattle give off different signals. So and yet, the Cercaria from Bovis has to get into a person in order for there to be an adult parasite of that species to hybridize with the other one.
Yeah, right.
So how does that work?
Well, I'm trying to understand. I don't understand how these cattle, sheep, and goats exist in Africa. So they were wild at some point. So these S. bovis were in the environment already, and they could potentially have infected humans along with a human strain.
So was Hematobium.
A long time ago.
Yeah, Hematobium was out there too. Although infected hominids.
Maybe when we increased the numbers of these animals, then the density of bovis in the water got higher, and that made it more likely.
And animal husbandry in Africa is an enormous occupation.
Yeah. So basically, this could have happened in the last 15,000 years.
Absolutely.
It doesn't happen.
Absolutely.
So it's as they say, if they can get more isolates, they may start to be able to date when this actual recombination occurred. Yeah. But now, Daniel, you picked this paper, I believe. So is there some medical reason why you did, Dickson?
It was Dickson. But I was going to say, the whole animal husbandry and when you're there in Africa, you start talking to people, you can get as excited about the animal husbandry as people here might, get about. And I have to admit, I almost bought a cow when I was in Uganda because completely irrational. What am I going to do with it? I had all kinds of plans where someone was going to take care of it while I was gone, and then I would come back and
adopt a cow.
Well, no, it was you buy it when they're young. That's really what you want to do. and then you sell it when they get to be full grown. I mean, a full grown cow is worth like $1,000, which is an enormous amount of wealth.
This is true.
And again, completely like, what am I going to do with the cow?
Right.
But you get excited. I mean, it is such a big part of the culture.
What I'm excited about is if this recombination has any effect on the biology of these parasites, of this Bocas,
I'll get back to your question, even though I didn't pick it. I was actually, I have to say, when I got to the conclusion here, I was like, oh, they're not really saying much. But what I thought was interesting is this concern that if you have these hybrids out there, and as we're seeing from this article, one of the things they're doing is they're maintaining the same tegument proteins and secreted peptides. So they're maintaining the same sort of invasion factors. Is there going to be more problems with these hybrids getting into the human population the way we saw these Senegal descriptions? Yeah. And will that impact our ability to treat and identify? And boy, if we get them out of people and we're thinking we're doing pretty well, but meanwhile you've got these hybrids coming back from our animals, it presents another eradication and control challenge.
Dickson, what's the band of the S. bovis in Africa?
That's a good question. If you look to see where the cattle are, I mean, there are maps that actually density map out where cattle are raised for each dot.
All right. So if a hybrid arises in a local stream, can it spread to other parts of Africa?
It depends on the migration patterns of the people who husband the animals.
People, they can bring them around.
They do.
And the eggs that they excrete can go back into the streams, and the snails can pick those up, right?
Well, the eggs have to hatch, and then the Miracidia seeks out the snails. But so there are other questions here that I would like answers to too, and that is the hybrids. How many eggs do they produce?
How fecund they are, right?
Yeah, exactly. And where do they live? Where do the hybrids live? Because does Schistosoma bovis live in the liver or in the mesenteric veins of the small intestine like Schistosoma mansoni does? Or do they live around the venous plexus like Schistosoma hematobium does in humans? I know there's an answer for this. I'm just ignorant of the life cycle of Schistosoma bovis. I don't know where the adults live. But if they're hybridizing with Schistosoma hematobium, then they're living around the venous plexus of the bladder. And that means that they're distributed not by feces, but by urine.
Mm-hmm. Okay.
Which is a more, let's say, compatible circulation with the earth, so to speak, because when you defecate in water or near water, that's one thing. But when you urinate and it gets into the water, it's distributed throughout the water column. It just goes all over the place.
Yeah. So I'll jump in, I guess, two things I'll say. So the Schistosoma bovis is mesenteric vessels, not vesicular, the way like a hematobium, right? So they're going to be, you've got your Japonicum and mansoni, that's like superior and inferior mesenteric. So this is going to be similar. So our mesenteric veins, not our vesicular. And then as far as distribution, which I guess it's sort of spotty, it's across sub-Saharan Africa. There are some descriptions in sort of Western sub-Saharan, but there's also in some Eastern sub-Saharan, like the South Sudan, Kenya type areas.
But Daniel, did you say where the hybrids live?
So the hybrids, we've just had the description in Senegal, right? So that's going to be Western.
No, I
Africa.
mean in the body of the infected individual.
Oh, yeah. So I can tell you where S. bovis is. I can tell you where the hybrids in humans, but I don't know where the distribution of the Schistosoma bovis hybrid.
Right, and that's critical for thinking about the epidemiology of this, at least.
Yeah,
right?
No, that is the important question.
The key here will be to see if the hybrids spread and become predominant, if they have some advantage, right? Or if this is just a rare thing that's localized.
Exactly right.
So that's the key. Then they say we have to get more isolates and see how widespread they are.
I was very excited when I saw this because I knew the earliest paper I could find on hybrids was back in 1988. They actually found some evidence for hybrids and hinted that this may be a problem, but this is the first systemic examination of that finding.
What they did not say is what this DNA is doing. Right? They say large parts of the genome are identical, but what is it encoding?
Well, they did start to talk about the antigens. Okay. And one of them was glutathione transferase that I'm pretty familiar with because I actually was in a group. I wasn't working on it, but they were. And it's an antigen which appears to be immunogenic and which appears to offer some protection against Schistosoma bovis in cattle. And they claim that it's likely that the S transferase for hematobium will be similar to the S transferase for bovis. And so therefore you'll have a vaccine candidate that might also be effective against hematobium as well. But there are other ones too. They have one for transpondin 2, which is an intramembranal protein, which we think is important for camouflage. And there are some other molecules in the integument as well that they mentioned that were candidates for vaccines. And so it would be great if you could vaccinate against Schistosoma bovis. Terrific. To keep this out of humans' environment. They have enough problems with their own species rather than having to worry about all these others. But
so we should point out that this stretch in bovis is identical to hematobium, but they don't know the direction, right? Because it's identical.
That's exactly right. That's exactly right. Who gave what to whom and how? So it leaves open a whole series of wonderful research proposals that you can imagine how this all happened.
So these, according to this article, they say that schistosomes have been targeted for elimination by WHO.
Yeah.
How are you going to do that?
Yeah, good idea. Yeah. Not easy. It ain't easy.
Is it by massive drug use?
Maybe praziquantel would be the logical choice here.
All right, Dinosaur Coprolites. They have
intriguing
trematode eggs. Water-dwelling dinosaurs.
Speaking of dinosaurs.
They walked in water.
They did. This is not on. This weekend, Friday of this last week, rather, I was in the Museum of Natural History with my wife and with her daughter and with our grandchild. And we get in the elevator to go down to the cafeteria from the 4th floor, which has all the dinosaurs. Mm-hmm. And who should walk into the elevator but Ellen Futter, the director of the museum. And I had a rare opportunity to introduce.
Did she recognize you?
Yeah, she did.
Did you tell her that we want to support TWiP?
We didn't go that far. I only had a brief
Did she say, "Oh, Dickson, how nice to see you"?
She did say that, actually, because I've been involved in some stuff there. And it was wonderful, though, to introduce her to my wife, who she had never met, my daughter, daughter-in-law, I should say, and my grandchild. Nice. All at once. And I said, "You just met the woman who transformed this museum from a dead animal visual stuff to a process." Right? You can go there now and find out about ecological relationships and things of this sort. And they've had some wonderful exhibits as a result of her
She was previously the president of Barnard, right?
That's correct. That is right. And she was delightful. Absolutely delightful.
Kind of like two Metacercaria, absolutely delightful. Is that right?
It's a little bit different than that, but yes, right. I would go there.
Shall we have a hero, Dickson?
We do, and this is one that Kevin alerted us to.
Because no, not Kevin.
It says Kevin writes.
No, John. Kevin sent in, well, I don't know which one you're reading.
Which hero would you like me to read?
Are you doing
Ann Bishop? Ann Bishop.
I thought we had done that.
No, we didn't do Ann Bishop.
Okay, fine. I can do Ann Bishop. Okay, John writes, it's long, but worth the read for your next hero segment. And so it's not long. It's very nice, as a matter of fact. So Ann Bishop. Ann Bishop, born December 19th, 1899, died May 7th, 1990, was an eminent British protozoologist and parasitologist. Bishop was born in Manchester, England, and developed a deep interest in science during her childhood. She enrolled at Manchester University in 1918, where she studied zoology for her bachelor's and master's degrees. During her undergraduate years, Bishop studied ciliates acquired from local ponds, producing her first major work on the reproduction of Spirostomum ambiguum. In 1932, she received her Doctor of Science from Manchester University for her work on the blackhead parasite Histomonas megalogritis. Bishop spent most of her career at the University of Cambridge as a fellow of Girton College. Bishop's research spanned a great number of parasites and antiparasitic therapies, but her best-known work is her study of malaria drug resistance. She produced a comprehensive study of Plasmodium, the malaria parasite, and of various chemotherapies for malarial disease. Her later study of drug resistance and cross-resistance in Plasmodium proved valuable to the British military in World War II. Bishop also discovered and named several parasites and protozoans. Bishop was elected as a Fellow of the Royal Society in 1959 and served on the World Health Organization's Malaria Committee. Bishop was also the main founder of the British Society for Parasitology, founded in the 1950s due to her keen perception that the field of parasitology would benefit if parasitologists cooperated more closely. She was initially given only £5 and a secretary to start the society. To raise the funds, Bishop passed around a pudding basin at the society's meetings. I wonder what a pudding basin is. Bishop was a frequent patron of the arts and a zealous cook, and she helped organize Girton College's Working Women's Summer School, an institution designed to provide continued education for women whose formal education ended around by the age of 14. Bishop died of pneumonia at 90 years of age after a short illness and was fondly remembered for her lasting devotion to science, her keen sense of curiosity, and her helpfulness and loyalty to her colleagues.
All right. Thank you, Dickson.
You're welcome.
How about a new case, Daniel?
All right. Hope everybody is ready. As a warning or a preamble, I will say that the next two cases are going to involve eyes. Okay. And so I'm just thinking what people can do is they can spend a lot of time really understanding which parasites might infect the eyes, how they might infect the eye. That'll help you get this first one correct. And then when I throw the second one at you, you'll be like, oh, this is just so easy now that I'm expert in this area of parasitology. We have a 9-year-old boy, and he's brought into a mobile clinic that was set up in a small indigenous village in a remote community in the Bocas del Toro archipelago. The story is that the boy initially had a red, painful eye a few months ago, and this lasted for 1 to 2 weeks. And now it's a few months later, and he's coming into the clinic. He had developed this problem shortly after the last mobile clinic visit. And so now he comes in. At this point, the eye is no longer red, but he has significant issues with vision in that eye. Fortunately, on this visit, we have an ophthalmologist with us who's able to do a careful eye exam. So initially, there's a note that there is some peripheral vision. The child can recognize hand movement, but there's an issue with any sort of sharp, clear vision. The ophthalmologist does a fundoscopic exam, and he notes retinal retraction and sees a quiet granuloma right over the macula of the affected eye. And so we're going to present to people what do we think this is, and then the other, and this is sort of a tough part of it, is let's say you had the opportunity to examine this boy back when he had the red painful eye. What might we have done, and would it have had any impact on this outcome?
All right. So this boy is living in a small remote village. Mm-hmm. What kind of a home is here? Is this a high-rise apartment building?
Yes, so it's a very palatial. Now it is a residence typical in this area. So it's there's dirt floors. Some of the homes actually have these sort of split boards where they're sitting up above. This is not a tin roof, so he's not living in one of the fancier homes in the area. There's a lot of animals around. I'll say no electricity, no running water, very primitive conditions.
And the water that they drink comes from?
So they collect rainwater, actually. There's usually one or two of the homes in the community, and this is sort of the initial sort of technological advance, is they'll have tin roofs, which then are used as places to collect rainwater. So it's very, very limited amount of water. Most of the people are only drinking about 6 to 8 ounces of water per day, and realize this is in the tropics. So very limited water access.
Is there any local water?
So occasionally there have been episodes where people have actually come in and done wells, but no, not in this community. It's just collected rainwater.
Mm-hmm. And what about for food? What do we eat?
So rice, beans, occasional fish, occasional
bananas,
plantains. Yeah,
plantains. No meat.
Very limited amount of meat.
Coconuts.
Some,
right?
yeah, some. Occasional supplement of fish. Occasional, and not very often. There might be chickens, might be pigs.
Pigs. I was going to ask.
And the
about
family
pigs.
consists of what?
So there are several children in the family. He's one of, I think there were 4 siblings that were still living.
Mm-hmm. And both parents are there as well?
So the mother brought him in. The father is off working.
And none of them have had similar experiences recently.
No, just the boy.
Only the boy.
Yep.
One eye only, right?
It was just the one eye.
Okay. Dickson, any questions? You know what it is?
I'm going to keep my guesses close to my chest until...
What are you, a poker player?
Yeah.
Okay.
So what I should recommend people do. Remember, I'm gonna do a couple of cases. So start looking into different fundoscopic exam findings and how they can steer us towards different parasitic infections.
Exactly.
And this is in Panama, right?
This is in Panama.
Only one eye involved, yes?
Yeah.
Only one eye.
Dixon, I have my eye on you.
And who do you have the other eye on? That's what Polyphemus said.
It did?
You know Polyphemus? He has the single eye on the tip of his...
Single eye? I thought it was a cyclops. Was that...
I think Polyphemus also has one.
Polyphemus, the giant son of Poseidon.
That's right.
With a single eye.
There you go.
It's pretty scary.
If you got one big one, you don't need more than one.
Well, you don't have binocular vision then.
This is true.
I want to read a few emails before we wrap it up.
Good idea.
In particular, we had a few cases ago an elephantiasis. What's the right name for that?
Yeah, that's it.
No, not elephant.
Did we decide not to call it that?
Yeah,
because
we decided not.
Wuchereria Banco.
Wuchereria Banco. What's lymph what?
Lymphatic filariasis.
Lymphatic filariasis. Anyway, so we missed Kevin's entry, but it's here in the notes if you want to look at it. And he says, take a look at the end. Let me see if there's something in the beginning here. I begin with a quote from the English surgeon Percival Pott, who many will remember elucidated the nature of the soot ulcer. Yeah, scrotal carcinoma of chimney sweeps, one of the first occupational illnesses to be clearly described. He also described tuberculosis, spondylitis, and developed many surgical techniques, among those being correction of hydrocele.
Right.
And he writes, hydrocele is so irksome a disease to the indigent and laborious, furnishes even the easy and opulent with such disagreeable ideas and apprehensions, and is to all who are afflicted with it so troublesome and inconvenient that every rational attempt toward relieving mankind from such an evil will, I make no doubt, be favorably received. So he has his usual copious notes, but he said, go look at the end.
Yeah.
Podoconiasis.
Right.
I was fascinated to learn about this condition. Wikipedia, podoconiasis, also known as non-filarial elephantiasis, is a disease of the lymphatic vessels of the lower extremities that is caused by chronic exposure to irritant soils in Africa and Central America. Terminal curiosity. Here we go. Several years ago, I visited a bedbound woman who weighed well over 500 pounds. In addition to the burdens of obesity, she was afflicted with massive bilateral lymphedema of the legs. This American woman had never traveled outside of the U.S. She had a condition known as elephantiasis nostrus verrucosa. This entity is reviewed by Castellani, a famous tropical medicine researcher who authored the 1913 note on copra itch referenced in TWiP 157. This is the only time I had encountered anything like the appalling photographs seen in tropical medicine textbooks. During our visit, I mentioned that her condition was known as elephantiasis nostrus. The patient immediately became very distressed by the association of her lamentable state with elephants. It took my best medical casuistry to backpedal out of this morass, and the whole episode taught me the profound subjectivity and emotional content of medical language as interpreted by patients. The WHO published guidelines concerning the naming of diseases in 2015. The press predictably branded the WHO initiative as a species of political correctness and reported the story in a slightly snide or frivolous way. As the above anecdote shows, a name can have profound emotional impact. Naming diseases after geographic locations can even have economic consequences. So he sends a list of diseases after various animals like swine flu, bird flu, lupus, ichthyosis. Who wants to be compared to a fish? Exactly. Moth-eaten alopecia, etc. Very interesting.
There's a disease known as Milroy syndrome that I used to use as a comparison to lymphatic filariasis. I don't know if you can find a reference to it there, but Milroy is, I saw someone on the streets of New York City with that, and I was told by someone that that's what that was, but pardon me, I'm not sure if that's the case. This man is obviously well-read and obviously curious beyond belief, and
wonderful.
He's amazing.
Go look in the show notes in the letters for the whole exposition. It's wonderful. Now, Anthony wrote that in TWiP 162, Dickson, you said over half the animals are parasites.
Yeah, I did say that.
There's one species, humans, that hosts how many parasites? Hundreds, presumably.
Hundreds.
We're not more infestation prone, just better studied. If that's so, then it's not just over half, but the vast majority of animals that are parasites. Is that correct?
I'm not willing to go that far, but I wouldn't disagree with that either. I have no data to back me up in either direction. But I know that there are some animals like crows, for instance. I've done many dissections of crows during a summer experience at a biological station, and we didn't find any parasites at all. Now, that doesn't mean they don't have them.
Of course.
But it means they don't accumulate them at the same rate we do. Now, remember this. The reason why people are so inundated with parasitic infections is because, let's just say this, we live everywhere, and we eat and drink everything.
Well, we're not everywhere.
Name a place that we don't live.
There's a remote island in the Pacific where
No. I'm sorry. We live there.
Okay.
We don't live in Antarctica, not permanently, and I wouldn't expect anything to be transmissible there anyway. But
most parts of Australia, nobody lives.
That's not true.
These huge expanses of land.
No. There are lots of Aboriginal peoples.
Nobody lives in my backyard.
Nobody. No, but you're being facetious now. We've covered the earth like Sherman Williams paint. Okay. And that's the reason why we've accumulated so many.
There are environments where there are no humans for miles and miles.
I know that, but there are lots of animals too, and plants too, that are specific for a region, and they will only accumulate certain numbers of parasites because of the geographic location. And so I don't think that everything is as heavily infected as humans. That's what I'm trying to say. Okay. And by the way, not all humans are infected with all of the species that you can't infect a human with. Like, for instance, African trypanosomiasis. We call it that because it's found in Africa.
Understood.
So you'll have to, modify that statement a little bit, but I would agree with you that it's well over half. I'm sure of that.
Anne sends a link to an article in The Atlantic. She says, a link on the importance of parasites. This is the sad story of a rare cat and its loyal parasite. We had scarcely discovered the parasite Felicola isidoroi when it vanished forever by Rebecca Giggs. And this seems to be open access. So you can read it. Thank you. And finally, from Anthony, you're doing a great job with science communication now, but perhaps through branding and a print medium, many more might be reached. And he sends a link to trueinfections.com. And if you go there, you see the cover of a magazine called True Infections, which I think Anthony must have put together. Because this is an old magazine cover, but it says on the top, was she HIV tested? And then they have some information about TWiP. So it's a little bit of a humorous.
Like True Confessions.
Yes. We need some print branding. Okay.
I like this cover. He said he didn't have sex overseas, but his mother was standing there when he said that.
Yes. That's great.
Oh, dear.
All right. That is TWiP 169. It is on all fine podcast players on your phone or tablet. Please subscribe. You get every episode. We see who's listening. We know you're there. No, it just helps us too.
We know you're there.
Now it's getting creepy.
We just need you to. We would like you to subscribe so we know how many listeners, not who you are. We have no interest in. Well, actually, we do are interested, but we won't find out if you subscribe. We'll just get numbers, and that helps us. And if you really like what we do, you can consider supporting us. You could give $1 a month. That would really help us. Microbe.tv/contribute. Questions and comments, case guesses, TWiP at microbe.tv. Dickson Despommier. No, not Dickson. I never start with Dickson. I start with Daniel. You sent me off with all the coughing.
Hey, I'm not doing that. It's my microbes.
Daniel Griffin is at Columbia University Irving Medical Center and parasiteswithoutborders.com. Thank you, Daniel.
Pleasure as always.
Dickson Despommier is at trichinella.org and fortimopolis.com.
There you go.
Thank you, Dickson.
Hey, a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. Music on TWiP is by Ronald Jenkins. Thanks to ASM for their support. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP
is
parasitic.